Hello, everyone, and welcome to today's webinar, Post-translational Modification Enrichment and Quantitation for Precision Medicine and Drug Development, presented by Dr. Connie Jimenez and Dr. Manuel Tzouros. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Cell Signaling Technology. For more information on our sponsor, please visit our sponsor tab on the left side of your screen and click on the logo to be directed to the sponsor website. Before we begin, I would like to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or you can report your problem by clicking on the Ask a Question box. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of your presentation window and follow the process to obtain your credits. I would now like to present today's speakers. Dr. Connie Jimenez, Professor of Translational Oncoproteomics, Department of Medical Oncology, and Head of the Oncoproteomics Laboratory and Director of the Proteomics Core, Amsterdam University Medical Center. And Dr. Manuel Tzouros, Project Leader in Proteomics and Mass Spectrometry at Roche in Basel, Switzerland. For complete biographies on our speakers, please visit the Biography tab at the top of your screen. Dr. Jimenez and Dr. Tzouros, you may now begin your presentation.
Thank you very much. So the title of my presentation is on cancer phosphoproteomics, and with this webinar I would like you to gain an understanding of the use of phosphotyrosine and global affinity enrichment workflows for label-free mass spectrometry-based phosphoproteomics, and we apply this in the oncology setting, and therefore I will also talk about our single sample integrative data analysis workflow, INCA, which is key for kinase activity scoring in single samples, and I'll highlight the way forward, how we envision it to be implemented in the clinic. So with this background, just to remind us about what is cancer, it's a devastating disease, and the big challenge is that that the genomes, cancer genomes, are massively aberrant, with multiple gains and losses over the genome, and basically every tumor, even if it's all colorectal cancers, each cancer has the individual aberrations, and therefore it's very challenging due to this high complexity and heterogeneity to predict based on genome data what the impact will be on the downstream cellular signaling circuits that govern growth and proliferation and oncogenesis and progression of cancers. And it's a huge challenge for the oncologist to determine what is the best treatment for an individual patient. Well, we know that a lot of the oncogenic drivers are protein kinases, and therefore there's over 30 right now, over 30 FDA-approved kinase inhibitors for the treatment of patients with cancer. But as already said, the drug response prediction based on genome data alone is not sufficient. For example, if a patient may have a EGFR mutation, but still not, that doesn't guarantee a response to an EGFR inhibitor. And since, of course, the proteome and the phosphoproteome is much more proximal to function, and these provide the activated drug targets, mass spectrometry-based phosphoproteomics offers an important complementary view of cancer, of tumors, and may be very important for selecting the right therapy for the right patients. Well, this idea, of course, is not new, and I would like to highlight here a great pioneering study, a large-scale study by the people from Cell Signaling, who studied in this 2007 paper published in Cell the phosphoproteome landscape in lung cancer, non-small cell lung cancer. They studied 41 cell lines and 150 tumors, and they came up with a very simple but effective ranking method. So based on spectral counting, phosphokinases were ranked, and what they observed was that whenever there is a genetic alteration that is an important in an oncogenic driver, that then this based on spectral counts, that this kinase is can be ranked is ranking high in individual samples, and this concept that they used to study the clinical, the patient tumors, and discovered that there was great heterogeneity in signaling. And in this pioneering study, new very important drug targets in non-small cell lung cancer were found using phosphotyrosine-based phosphoproteomics. And this landmark study inspired me to really focus on phosphoproteomics and implementing that in the lab as a way to stratify patients for targeted therapy with kinase inhibitors. Since then, we have been successfully using the workflow. So this is just we do everything label-free, and this can be done large scale, like was done in this pioneering study. So we have now lots of projects in lots of tumor types where we answer clinically relevant questions and biomarker development using this, especially this phosphotyrosine phosphoproteomics workflow, but we also use global phosphoproteomics. So this is just the background. So now a little bit, I would like to share a little bit more on the sample preparation that we do. And here we and what is well, yeah, so the phosphotyrosine immunoprecipitation workflow using, we use the cell signaling PY1000 antibody that is conjugated to agarose beads. And in our hands, the protocol works really well at a 5 milligram protein input level and can be downscaled even to the 1 milligram protein input level, which is compatible with tumor needle biopsies. Well, when you work with cell lines, the general remarks is to make sure the confluency is constant across experiments, because confluency, of course, influences signaling. And it's very important to practice the workflow before you do a real experiment with different groups or samples, and make sure you organize, you work in a really organized and fast manner. Since phosphorylation can change rapidly, and you don't want pre-analytical variables influence your results. And also when we do everything label-free, because that's advantageous when you want to go large-scale clinical sample profiling studies with multiple groups. So we make sure we always have an alternating study design, where if we have, for example, two groups, that we alternate cases and controls to minimize the influence of different factors. And in this way, we know we can do label-free proteomics at large scale. So here we, just the steps of the protocol. So we start with a lysate of cells or tumor samples. We digest proteins to peptides. We desalt using C18 SEPA cartridges. Then we lyophilize the sample, and then it's ready to do the phosphotyrosine immunoprecipitation step. After that, there is another stage tip desalting step. And then we typically analyze the phosphotyrosine enriched peptide fraction in a 2-hour nano-LC-MS run. And here we see the performance of replicate samples. So it depends on the cell line how many peptides you get. So some cell lines have a lot of peptides. So in between, let's say 500 and 1500 peptides we get. And this is here we see the data for our reference cell line HTT 116 that we always take along in every sample. So we in every experiment. So we know this cell line really well. And we can do the capture at very high, with high reproducibility, with highly correlated spectral counts. And we have spent quite some time to trying to improve on this protocol. And our idea was, if we get rid of the non-phosphorylated peptides, because there is a background of non-phosphorylated peptides. If we can get rid of them, then maybe we can even retrieve more phosphotyrosine peptides. So we try to use detergents to do some cleanup, but that did not really improve it. We have the performance was really stable relative to the default protocol. We also tried titanium oxide, pre-enrichment of the global phosphoproteome, but that also did not improve results. We did not get higher yields. The only difference that we can see is the difference between generations. So overall, the default protocol works already optimal. That is our conclusion. And when we have the spectrometer Q-exactive HF, so apparently the instrument is already fast enough to analyze those peptides, and further enrichment doesn't help. Here just for comparison, so typically we like to do both enriched separately for the phosphotyrosine-based phosphoproteome, and then on an aliquot of the unbound fraction of the phosphotyrosine item, we do a global phosphopeptide capture using titanium dioxide. So typically we do 2 phosphoproteomics runs to get a good insight into the phosphoproteome of a sample. And here we also see the performance, the Tiox-based capture, and where you can see it can be done reproducibly. So up till now we have now close to 2,000 phosphoproteomics MS runs with over 12 million identified phosphopeptides MS spectra, over 140,000 unique phosphopeptides that map to total 437 protein kinases. So just in our lab from the past years, we have observed 82% of the phosphokinome, and in total 11,000 phosphoproteins. So these are the numbers. So now a few words on phosphoproteomics analysis of clinical material. In our case, these are tumor tissues. So a guideline is that after tissue lysase, the protein yield 1 to 10 of the wet weight. We like to make sections, fresh frozen tissue cryosections, to ensure fast lysis of the sample. And here you can see the numbers of how many slices we typically use as input, about 20 slices of 10 micron thickness. So then we get a very quick lysis, and we use tissue to lysis buffer ratios around 1 to 20 to 1 to 40, and we always do some test lysis because we always want to get this right, because this is very important for protein yield and reproducibility. So you don't want to use too little buffer and then get low recoveries and naughty slimy samples. And so we always, for each new tissue type, we always test this to have the optimal rate. So the general remarks when doing a phosphoproteomics experiment, especially when you want to do this large scale, we keep the samples frozen until the last moment. We thaw tissue lysates one by one to avoid samples sitting a long time on ice and waiting to be processed. So we really try to avoid that, and we lyse them sequentially, and then we do quickly the sample prep. And well, if we have tissue cryosections, we typically do not even need a potter, but for tissues that are a little bit tough, a potter device is handy. And before we freeze the lysates, we always make some aliquots. So a big aliquot, of course, for phosphoproteomics, and we also make a few small aliquots for phosphotyrosine Western blot analysis, and also for a global protein staining to assess sample quality. So tumor needle biopsies yield enough protein for the phosphotyrosine workflow, and we typically whole biopsy for this analysis, and it yields between 1 to 3 proteins. So here we see the performance. We sort of took it to the test, taking multiple needle biopsies from the same tumor, and when we do an unsupervised analysis using phosphoproteome data, we see that needle biopsies taken from the same tumor, that they cluster together, as you can see in the colors. And what you can also see is that every colon tumor has its own phosphorylation profile. So you can imagine that patients, if we were to select an inhibitor patient, that they would get different selected, matched on the individual profiles. So phosphoproteome data are very complex. That these are large data sets, and to make this practical, so we have been thinking what kind of data analysis can be done. Because in the end of the day, if an oncologist is to use this data, we need to boil it down to a simple report, and this is what inspired us to make a dedicated data analysis pipeline that can enable phosphoproteome-guided kinase inhibitor selection based on the profile of a single sample or patient. And what we came up with a method that makes use of all the data resources out there. So we use the phosphokinase data itself, a la Ricova paper, the Cell 2007 paper. So we rank on the phosphokinase data, but we also make use of the kinase substrate relations to inform us about upstream kinase activity, and we make use of the kinase motif data from network-in. So basically what we do is we take the phosphopeptide data to do a ranking on the kinase observations and the activation loops, and we use the phosphosite data to link to upstream kinase activity using Phosphosite Plus database and network-in. And then we combine those the kinase observations and the substrate observations via this formula. It's a product, so that means that there has to be observations on both sides, kinase and substrates, before we say a kinase was active in a sample. And we have 3 different outputs, just a simple bar graph ranking of inferred kinase activities, kinase substrate network. And this workflow was recently published in Molecular System Biology, and we build a website where people can upload their data and it is available. This is just to show you that, so we benchmark this workflow on a number of cancer cell lines with known oncogenic drivers, with mutations or amplifications. And what we see is that using this INCA ranking based on kinase and substrate data, the oncogenic drivers, they really rank high in the individual cell lines highlighted here, the drivers. So in this way, it works. And also over here we have a lung cancer cell line where there is mutant ALK, but you can already see that there's other kinase activities that are quite high. There's not this outlier behavior. And over here we can look at one example of the kinase substrate network of a CML cell line that has hyperactive ABL due to ABL fusion. And we know that if we target this kinase with drug, that the cell line dies. And here we have an example of a resistant cell line. So here we have the mutant ALK in this lung cancer cell line, but you can see that there's a lot of other hyperactive kinases, and we know that this cell line is resistant to single drug ALK inhibition. And the indication here is that we need more than one drug to. And the INCA analysis gives suggestions for other kinases target in this. So that brings me to the last question. Can we predict which kinase inhibitor to select based on a baseline INCA profile? And to answer this question, we analyzed patient-derived xenograft tumor models for colorectal cancer, and we analyzed tumors from a well-characterized cohort from Italy. We collaborated with Livio Trusolino, and a few of these models based on genomics, they didn't have a clear genomic aberration. And we asked the question, can we use INCA for target selection, genomically unexplained resistant DX models for colon cancer? And here we see two INCA profiles of two xenograft tumors, where we see a relatively high activity of insulin receptor and some other kinases. And to test whether these provide alternative targets for treatment, we tested this in organoids, and we first verified using the phosphotyrosine immunoprecipitation workflow coupled to INCA analysis that the organoids preserve the signaling from the original tumor, and the way we select the optimal kinase inhibitors is we look at the whole INCA profile, and we match it with other resources where we know kinase inhibitors often have their targets and off targets. These drugs are quite dirty, and we make use of that to do an optimal matching of kinase inhibitors with their target space, and we map it onto the target space, the INCA profile. So for example, the BMS inhibitor, insulin receptor, but also other kinases highlighted. Does it work? The answer is yes. So high-ranking INCA kinases targeted by the drug give growth reduction of the organoid, or viability reduction. And when we combine two inhibitors, we get even much more viability reduction. And targeting a low-ranking INCA kinase is able with immunology doesn't work. Very important to target high-ranking INCA kinase. And this brings me to the end. The way forward, we see that phosphoproteomics will be part of a multi-omics precision medicine pipeline, where we will use mass spectrometry to generate phosphoproteome-wide single assays as a single readout to determine kinase hyperactivity relevant for cancer. And it doesn't matter whether hyperactivity was due to mutation, amplification, gene fusion, microenvironment activation. Phosphoproteomics provides a great downstream readout of oncogenic hyperactivity. Phosphotyrosine workflow is very important because it provides a high throughput view of clinically relevant kinases, and the Inca-based sample patient report with rank kinase activity is very important for drug prediction. And with this, I come to the end of my part of the webinar, and I like to thank all the people, clinical collaborators, and Dr. Tzouros.
So hello everyone. Now it's my pleasure to walk you through the second presentation of this afternoon's webinar. And I would like to present you on a project where we automated K-e-GG peptide affinity purification. And we did this with the intention of applying to the identification of E3 ligase substrates. Just a quick outline of the presentation. I'll start with a brief introduction, and I'll follow with a description of this, the way we automated the K-e-GG peptide affinity purification. Then I'll show you an application we had on defining the substrates of the E3 ligase CBLB. And then I'll conclude, briefly mention the next steps we are currently working on, and also briefly acknowledge the people who did the work. Protein ubiquitination is actually a multi-step process that involves E1, E2, and E3 enzymes. And just to mention, in humans, there are 2 E1s, a bit less than 40 E2s, more than 600 E3s, which can basically ubiquitinate thousands of substrates. These substrates usually get ubiquitinated on lysine residues, and they can be mono as well as polyubiquitinated. And this is because ubiquitin itself contains 7 internal lysines. These different ubiquitination architectures, they lead to different signaling outcomes. And probably one of the most famous one is proteasomal degradation, but it is also involved in protein trafficking. For proteasomal degradation, that's basically what you have in the figure on the left-hand side of the slide. Now, the ubiquitin proteasome system, or UPS, is actually a very interesting system for the pharmaceutical industry, because it can be used to degrade disease-causing proteins. And this can be done, for instance, with the use of a degrader. So a degrader is a bifunctional molecule that includes both a target protein and an E3 ligase ligand. And these two parts are separated by a linker. So as you can see on the second figure on the left-hand side, so a degrader would be this multicolor molecule that you see, and it basically recruits the ubiquitin-loaded E3 complex in blue. It recruits the target protein in green, and basically catalyzes the ubiquitination of the target and forces this protein to be degraded. And in this whole context, it is important for us also to explore substrates of these E3 ligases and their use as potential target engagement biomarkers. The study of ubiquitylome on a global scale is helped by the fact that cell signaling offers an anti-K-GG ubiquitin remnant antibody, and this is something you can use in the following context. So usually ubiquitinated proteins, if you digest these proteins using trypsin. So trypsin is an enzyme that specifically cleaves after lysine and arginines. So all ubiquitinated part of a protein will basically generate a peptide that contains an internal lysine that is modified by a diglycine residue on the amino side chain group. And using the K-GG ubiquitin remnant antibody, you can basically enrich for these species. And this you can do in a whole cell extract that has been digested with trypsin. After affinity purification and elution from the beads, you can analyze the samples by LC-MS, and then use softwares to identify the peptides, and also do some further bioinformatics or relative quantification steps on it. And here I just wanted to measure basically the first report in the literature that is a landmark paper using this technology that was published in Molecular Cell back in 2011 by the group of Kim et al. Now, this global analysis of ubiquitilomes and these antibody tools, they also come with a certain number of challenges. And first of all, ubiquitination is a low abundance post-translational modification, which requires large amount of starting material. Typical recommendations there are to start with at least 10 milligram of protein or peptide sample, but you can see reports where people even go up to 50 milligram. This is of course limiting for certain sample types, for example primary cells, or when you have larger experiments where you want to compare different conditions and replicate. I also have to mention that the use of these PTM scan kits, they require a bit of training for typically new users until they work in a nice and reproducible manner. So having these challenges in mind, and also due to the fact that in the last 10 years or so, there were some improvements made or proposed for this type of approach, and namely the group of Udeshi showed that you can use this workflow in a high sensitivity manner by lowering the antibody amount that you are using per IP, as well as cross-linking the antibody to the solid matrix. Colleagues from cell signaling technologies, led by Matt Stokes, also reported that you can run basically one of these PTM scans in an automated way using an assay map Bravo from Agilent. And more recently, the Chris Rose and colleagues showed that you can combine such a K-GG enrichment experiment with the tandem or TMT mass tags technology when you want to do multiplex quantitative experiments. So with these different inputs in mind, we optimized an automated K-GG peptide enrichment workflow, which is basically summarized on this slide. So we use it for cells or tissue, and we can do that in principle from 1 to as many samples as we have. I mean, typically we're in the range of 20 samples. Proteins are extracted. We we lyse the cells, we digest the proteins with trypsin, and dissolve them by solid phase extraction. Then we take some protein A cartridges where we immobilize K-GG antibody. And here there's a mistake actually on the slide. It's not 31 milligram that we immobilize, but 31 microgram. After immobilization of the antibody, we cross-link the antibody to the protein A cartridge using dimethyl pimelimidate, or DMP. After this is done, we perform the affinity enrichment step of the K-GG peptides, and this works down to 1 milligram of input material. After that, we elute our K-GG peptides using 0.5% TFA. And in case we have a multiplexed experiment, we can TMT label our eluates, combine the samples together. If we need deep coverage of the samples, we then offline fractionate our samples using pH10 reverse phase. And we measure our samples by LCMS on an Orbitrap Lumos. And for this, we use typically synchronous precursor selection method, or SPS, to get accurate quantification. The data we process using the Proteome Discoverer software and Mascot as a search engine. So one of the first things we wanted to assess was basically the effect of cross-linking the antibody to the protein A cartridges with DMP, and what this would bring as an improvement. So we did an experiment where we took Jurkat T cells that we pre-treated with MG132, which is a proteasome inhibitor, and just guarantees that the ubiquitinated proteins, they accumulate in the cells, and the K-GG peptide signals are high enough that we can detect something. And in this case, we used 2 milligram peptide input material. This is just for the note, it's one of the only experiments where we used 2 milligram material. Most of the other cases, we used only 1 milligram. So we basically run 2 of these samples using a non-cross-linked protein A cartridge and a cross-linked one. And after LCMS analysis, we basically got a 5-fold increase in K-GG peptide group identification. And this came along with an increase in the total number of peptide groups that could be identified by 26%. And this also comes along with an increase in the number of MSMS the instrument performs. What is also important is by using cross-linking, we also increased the selectivity for the K-GG peptides, and this went from 7 to up to 28%. So there's really a benefit in cross-linking the antibody to the cartridges. Then we did the following experiment. Of course, automation should basically bring you a more robust workflow with less variability in the data. So we wanted to assess that. And for this, we performed 2 experiments side by side, where we took Jurkat cells and 5 replicate cell population were treated with DMSO as a control, and 5 others with MG132. And these 10 samples each were either run on the AssayMap Bravo with the automated K-GG affinity purification workflow, or using a manual bead-based approach. And here I have to mention that we used protocol as described in the paper from Udeshi. After running, then the eluates were TMT labeled with a 10-plex kit, fractionated and analyzed separately by LCMS. We then plotted the log10 abundances of the reporter ions for both treatments and plotted the data of the automated versus manual data side by side. And then you can see them here color-coded by orange and blue. And what you can see is that we have a reduced variability when we use the automated workflow. As you can see, the boxes and the whiskers are smaller, and we also get less outliers. And this is actually more pronounced for the MG132 treated samples compared to the control one. Next, we wanted to assess if we can basically reuse these cartridges. And I think this is a cool experiment that we performed. Basically, we prepared two cartridges that we loaded with 31 microgram and not 31 milligram of the K-e-GG antibody, cross-linked with DMP, and we used again Jurget cells, 1 milligram inputs in duplicate, and enriched them with these two cartridges. And then we repeated that for 5 times in total, using always fresh sample. Then the 10 eluates we had, we also TMT labeled that, fractionated and analyzed by LCMS. And here again, we plot the log10 abundances of the reporter ions, and basically you can see that there's no observable decrease in performance. So these cartridges can be reused. Maybe I should mention here that this is true for a certain amount of time we tried. So all these 5 IPs were performed within 2 days. Now I moved to an application of this workflow for studying the ubiquitin ligase CBLB, or Casitas B lineage lymphoma proto-oncogene B, which is a ring-type E3 ligase and is a known negative regulator of T-cell receptor signaling. It is also involved in T-cell activation control, peripheral tolerance, and exhaustion. Degrading CBLB with a degrader that I mentioned earlier is an attractive approach in cancer immunotherapy. And of course, what would be wishful to have are basically substrates of CBLB that could be used as target engagement biomarker during the whole drug development and optimization process. So for this, we performed the following experiment. We took Jurget T cells that we treated with an siRNA against CBLB, and also used a scrambled sequence as a control siRNA. And we also added suboptimal T cell receptor activation by treating these cells with an anti-CD3 antibody. We performed the whole workflow that I described before, and we also actually took a portion of the samples to perform proteome quantification. After this, we managed to quantify more than 8,000 K-e-GG peptide groups and 7,500 proteins. Looking at the CBLB itself, with this we managed to knock it down by 40% in this experiment. Now, looking at the K-e-GG data set, we highlighted 2 interesting tyrosine protein kinases, SAP17 and LCK, which had several lysines that were identified as being modified by ubiquitination and also modulated by CBLB. And this was actually quite interesting because these two tyrosine kinases are known interactors of T-cell receptor. Furthermore, the two K-GG sites, so K484 of Sub17 and K379 of LCK, are located in the kinase domain and are upstream of regulatory tyrosine sites. So these are two proteins that are still in further exploration in the oncology team. With this, I would like to conclude. So we basically optimize the workflow where we load 30 microgram of K-GG antibody on protein A cartridges followed by DMP cross-linking, and this allows us to automatically enrich for K-GG peptides down to 1 milligram of tryptic digest using the assay map Bravo. In combination with TMT10 or 11-plex plus pH10 reverse phase fractionation, it allows us to quantify up to 8,000 K-GG sites. Now this automation comes with the fact that it should be operator independent and also shows reduced sample-to-sample variability. A nice thing is that you can basically reuse these cartridges. And then we applied the workflow successfully to the identification of substrates of the E3 ligase CBLB. Just as a next step, I wanted to mention that we try to use the exact same workflow with different motif-specific antibodies, and currently we try out to use the antibody against methylated arginines. We also try out to use biotinylated motif antibodies in combination with streptavidin cartridges to do the enrichment, and this works also pretty nicely in our hands. So last, I would like to thank a couple of people from our department, in particular Jonas Colibius, who was a master student with us for 9 months, and basically he's the one that generated all the data I showed you today. Also, thanks to the oncology colleagues for the collaboration on CBLB. The people from Cell Signaling Technologies, Matt and Chuck, for providing some advice on how to use the K-GG antibody, and finally Sriram and LabRoots for the opportunity to present during this webinar. And with this, I hand over to Christy for the Q&A session.
Thank you, Dr. Tzouros and Dr. Jimenez, for your presentation. Yes, it is time for our live Q&A. Now, if you have a question you would like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. Okay, let's get started. Dr. Jimenez, let's start with this question for you. Can the phosphotyrosine IP protocol be combined with Tiox capture on the same sample? And if so, which order do you perform the capture?
Yes, so the answer is yes, it can be done on exactly the same sample. So typically we start with the lysate with the phosphotyrosine capture, and then we use an aliquot of the unbound fraction of that capture for the titanium dioxide based capture. And we also, for protein expression, we typically, one can also do protein expression profiling on another aliquot of the unbound fraction, but typically we do that on a fresh tiny aliquot of the initial sample.
Thank you, Dr. Jimenez. Dr. Tzouros, let's jump over to you for this question. Did you try other PTM scan motif antibodies, such as the phosphotyrosine, with your protocol on the assay map Bravo?
So yes, as I was mentioning in the conclusion slide, we try currently to use the antibody against methylated arginine. We just very recently got some data. It seems to work. We just maybe have to optimize it for this particular type of post-translational modification. The phosphotyrosine itself, we never tried on the assay map Bravo so far.
Maybe I can add to that.
Sure.
Can I? Yeah, so I know that people at Cell Signaling have also successfully used the SAMAP Bravo platform to do the phosphotyrosine immunoprecipitation, and they use 2 milligrams of protein for that. And actually, we are purchasing right now the Bravo platform, the SAMAP platform, in order to automate our phosphotyrosine IPs and other captures on there.
Thank you. Now, Dr. Jimenez, I believe this one's for you. Now, with optimization of current global phospho-enrichment methods like TIOX and IMAP, and speed of new MS instruments like QE-HFX and timsTOF Pro, why would you need phosphotyrosine enrichment, since it requires high input, milligrams protein lysate, complex and lengthy workflow, and is expensive?
All right. Well, let me start by saying that the phosphotyrosine part of the whole phosphoproteome is only about 1%. So even enriching for that 1% is always better than a global method. Because also if one would want to achieve with the global method the same performance as with the phosphotyrosine workflow, which I think is difficult, then you would need to really start out with a very high, way higher starting amount, and do a lot of fractionation to sort of get close to the depths that one can get from direct phosphotyrosine capture coupled to single shot MS. So it's very challenging, if not impossible, to reach the depths of the phosphotyrosine-based phosphoproteome in a global experiment. So that's why we perform two different captures, like consecutive or sequential captures, do two runs to get the best of both worlds.
Thank you. Okay, we have another question coming in. Do you have to do a buffer exchange between phosphotyrosine and TIOX enrichment?
We have a SEPAC step, a desalting step in between. SEPAC cleanup. So the answer is yes, there is buffer exchange.
Thank you. And we have another question coming in. It's a little lengthy in a few parts. I am working with lysine methylation, which is also very low abundant as ubiquitination. I guess that after the immunoenrichment, you get very low amounts of peptides, and I'm scared of losing a lot of them doing HPH fractionation. Is this not the case? And isn't the sample after the immunoaffinity already much less complex? And did you get many more identifications with the extra fractionation step?
I can take that one. Actually, the very first trials we did were direct measurement of the eluates after the IP. So we didn't try to combine with high pH reverse fractionation yet. I don't really know if this brings anything, but it's true that the numbers of methylated peptides you identify then is really in the range of hundreds only.
Thank you, Dr. Tzouros. Now, Dr. Jimenez, do you foresee that phosphoproteomics will be part of clinical diagnosis in the hospital in the near future, or do you believe it will remain in research only?
Well, I do think it will have eventually a place in the clinic, but we need to. We are not there yet. So currently, everything we do is in the research context, applying phosphoproteomics to tumor needle biopsies collected in clinical trials, and see what the added value is directly one-to-one in comparison to genomics. And of course, we need to we need robust workflows. We need to automate. I think mass spec will be the preferred readout, and in this context, I think data-independent acquisition mass spectrometry is of interest there. So this is also something that we are working on phosphoproteomics using DIA-MS, and it takes, of course, close collaboration with clinicians and pathologists and that's so. I cannot say when this will happen, but I hope that we have strong proof of concept in the coming 5 years, and then maybe in 10 years from now, it will be as normal to run a phosphoproteome profile along with a genome profile in the context of advanced cancer.
It looks like we have time for one more question. Dr. Tzouros, we'll have you close out with this question. How efficient is the K-GG peptide affinity purification with your protocol?
Actually, to assess efficiency, we tried to synthesize heavy labeled K-GG peptide standards and spike them in a sample at the beginning of the workflow, and see how much we would recover after the experiment. The one trial we did didn't look actually to be very efficient. We recovered something like 10% of the peptides. I think that's about what you can get with this workflow. However, we need to repeat this experiment to get more confidence numbers there.
I would like to once again thank Dr. Connie Jimenez and Dr. Manuel Tzouros for his time today and her time today, and for their important research. We would also like to thank LabRoots and our sponsor, Cell Signaling Technology, for underwriting today's educational webcast. Now, before we go, I would like to thank the audience for joining us and for their interesting questions. And as a reminder, questions we did not have time for today and those submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Thank you for joining us, and we hope to see you again soon. Have a great day.